These are the notes on RNA, transcription, and translation. You should have gotten the note page from the chapter 13-14 drawer. If you didn't, go get that now. We are not doing the entire set of notes on this video. We're going to stop after doing transcription, so we'll do RNA and transcription. We'll practice transcription a little bit, and then you'll watch a different video to do the translation part. I like to separate those because they're very different. But we're going to start with what is RNA, and I've got RNA's name here. You might want to write it down on the top of your notes, ribonucleic acid. So RNA is ribonucleic acid. That's a little different than DNA. DNA was deoxyribonucleic acid. And those names, they're named based on their chemistry, which is something that honestly I don't know fully. I know it's a sugar. But the focus is different names, they're going to do different things, but they're both nucleic acids. So what is RNA? RNA is just another nucleic acid that's going to help us make a protein. Remember, DNA held our information. And if you remember, for us, DNA is located where? It's in our nucleus. Well, RNA has to help us make protein. Protein isn't made in our nucleus. But I don't want my DNA to leave the nucleus either. So I'm going to need RNA, and we're going to talk about this a little bit more. But remember, what do we use protein for? We use protein to build up some of our body. We use it for our muscles, our immune system, our hair, and our nails. Remember, it made a lot of stuff for us. So we need to be able to make protein, not just sometimes, all of the time. Think about it, you're making at least your hair and your nails all the time. So we need to be able to make protein all the time. And that's what we're going to use RNA for. So there's going to be some similarities and some differences. We're going to focus on the differences here. There's 4. The first difference is that RNA is ribose, while DNA is deoxyribose. So it just means that RNA uses a different sugar than DNA uses. Remember the nucleotides were a phosphate, a sugar, and a nitrogen base? RNA is the same thing. It's still phosphate, sugar, and a base. It's just a different sugar. Instead of being deoxyribose, it's just regular ribose. We don't need to know much more beyond that, but it's just a different sugar. So that's number 1 difference. The second difference, RNA is single-stranded, meaning that it only makes half a ladder. This is very different from DNA with the double helix. Remember, DNA was 2-sided, while RNA was 1-sided. So RNA is only going to have half a ladder. It literally looks like a ladder's been chopped right down the middle, and it just has one side. This is very different for us.
You're early.
Sorry guys, back at it. So RNA can leave the nucleus. It says RNA is can. One of those. RNA can leave the nucleus. This is because it's single-stranded. So RNA can go outside the nucleus. DNA is stuck in our nucleus and it can't leave. Partially because it's too big. Being double-stranded makes it harder to leave. But also, it's really important that it stays where it belongs. Think about your brain. Your brain has all of your knowledge. Do you want your brain oozing out of your head and going to other parts of your body? I don't, right? I need my brain in my head. If I want that information, I use nerves and I use electrical impulses to move it around my body. Well, DNA, we don't want that oozing out of the nucleus. It needs to be stuck in the nucleus. So when I need information to leave the nucleus, I use RNA because RNA can go in the nucleus and can come out of the nucleus. So RNA is made in the nucleus and then it can leave. And that's good. Because remember, our proteins are made at the ribosomes. Our ribosomes are not inside our nucleus. So in order to get the genetic code to the ribosomes, we need to make RNA that can take it there. So it's really important. One of the most important differences is the fact that RNA can leave the nucleus. And the last one that we're going to look at is that RNA uses uracil instead of thymine as one of the nucleotides. So instead of using T, it uses U. It uses uracil. And that's, just one of those random differences. It's gonna change things just a little bit, and you'll see what I mean. But focus on that for me now, that it's U and not T. So remember, we said this already, RNA stands for ribonucleic acid, and it's going to have 4 nucleotides. The G, C, and the A stay the same. We still have guanine, cytosine, and adenine. And the last one there is U, uracil. I added the phonetics there so you know how to pronounce it, uracil. So now we have GCAU. So we're not GCat anymore, we're GCow. This one was a little bit harder to find cool pictures of cows. You know, not many people dress their cows up like gangsters. I don't know how to help that. Not quite the same as when they dress up cats, apparently. But it helps me remember GCow now instead of GCat. And again, just like before, the G and the C go together, and the A and the U go together. So we're still got our pairs, and now we've made a GCow. You probably think I'm weird, but I love it. So what do we do? How do we make RNA? So RNA is not just found in our cells. And that's because RNA's got a really specific process. RNA is supposed to make protein for us. We don't just keep it sitting around. We have to make RNA. And we make RNA through a process called transcription. And here's the definition of transcription. You might want to start it. Transcription is taking DNA and making RNA from it. So we take our DNA and we make RNA. Why are we going to do this? Remember, DNA codes for everything we need. But we need that information to go out to the ribosome. And the DNA is stuck in the nucleus. So we make RNA so that we can send information out from the nucleus, and then it can get to the ribosomes and make protein. Because we want the information from our DNA going out there. We just don't actually want our DNA itself leaving the nucleus. So we make RNA. It's not hard to make RNA. It's just different. So just like we made replication, it wasn't hard. It just took a minute. Transcription is just going to take a minute. So you're going to open the DNA, just like we did on replication. First step, open the DNA. We're going to open it, spread it out. We use the left side of the DNA to make RNA. Always, always. You use the left side. You're going to match the DNA with its opposing RNA pair. That's going to be the hardest part. And then you're going to release the RNA and close the DNA back up. So from beginning to end, our DNA should not change. In step 1, we should have a piece of DNA. In step 4, we should have that original piece of DNA back. It sounds silly, but it's really important to redraw the DNA again because we have to prove that our DNA went back to normal. And then we're going to make the RNA. Now there's a practice on here that we're going to go through in just a minute, but remember this part here. G is going to go with C. So if there's a G in DNA, I'll turn it into a C in RNA. If there's a C in DNA, I'm going to go with a G in RNA. T still goes with A, and it's this last one that's different. A now goes with U. Think about it. A should pair with T, but there is no T in RNA. So when there is no T, I have to use a U. So A turns into a U because it can't turn into a T. There is no T to turn into. Let's look at this. So we're going to, remember, open up, use left side to make the RNA, match the DNA with its RNA pair, and then release the DNA and close it back up. Here is your example for the steps 1, 2, and 3. So you see here that I have my original piece of DNA. I opened it up. See, I broke it in the middle and opened it. I've taken the left side and I've made an RNA pair against it. I've used a second color here, and you want to use a second color when you make the RNA. See, DNA was in the white, RNA is in this green. So I took my DNA left side and I looked. I said, okay, G turns into C, G turns into C, A to U, T to A, T to A to U. So I've made the DNA. I want you to come over here and do step 4. Step 4 is just writing the RNA, so writing the green again, and then putting the DNA back together in its original piece. I want you to do step 4. If you're not sure, if you look good, call me over, let's look at it. But for step 4, you just rewrite the RNA, that's the green here, so you'll rewrite it, and then close your DNA back up to the way it used to look. Draw this whole thing down at the bottom of your page where it says practice transcription. Make sure you're labeling step 1, 2, 3, and then you're going to label step 4. When you're done this, move to the next one. It's going to give you another practice. Go ahead and practice this piece of transcription too. You're going to need a second color. I want your RNA in a second color, your DNA in your original color, maybe your pencil, whatever you're using. But your RNA should be a different color. Go ahead and practice this transcription on your notes. Call me over if you need me to look at it, if you're not sure, if you just want it checked, because your next assignment is just doing a bunch of transcription because you need to practice with it. So this is where the notes end. I know there are more words, but we will get to those after this. That'll be translation now. It's something different. So stop here. If you need to, go back and listen to the video, read back through your notes, or ask me, and we'll make sure that everything's understood before we move on.